You're looking for the best exome that exists, and you can learn more about this in our webinar. Welcome to our webinar on CeGaT ExomeXtra. Today we will look at the question, what is the Xtra in our exome diagnostics? I'll start with a brief overview. ExomeXtra, as any exome diagnostics, starts in the wet lab, where we generate very high quality sequencing data from your samples. This means that we have to think about every step, optimize the protocols, and of course also continuously make sure that we keep the data quality high. We deliver this data to our bioinformatics team, where we use proprietary data analysis algorithms to produce the optimal results that our medical team can then analyze. And of course, we have very qualified medical experts that look at this data and then write a medical report that is clinically useful for you and your patient. So these are the three main steps, and they are all done in-house at CeGaT, and we have years of experience. But there's another step that is often overlooked, and that is content design. Content design answers the question, what should be in the exome in the first place? The definition of an exome seems to be very clear. An exome is all exons of all protein coding genes in the human nuclear genome. Sounds easy, but there are a few questions we have to answer when we want to use an exome in a clinical diagnostic setting. What is included in all protein coding genes? Are there other regions in these genes that we want to look at? Are there maybe other types of genes that are interesting? Are there other genomic regions we should assess? And also maybe there are other genomes that we should look at. We will look at all these questions one by one. Let's start with what is included in all protein coding genes. There are different databases that define the human exome, and they give different answers about the size of the exome. So as you can see here, the 5 most used current databases vary in size for the human exome between about 31 megabases and about 35 megabases. And if you use an off-the-shelf exome, you cover a decent amount of these regions. CeGaT ExomeXtra, because we use current databases, covers more of the databases. And as you can see, we have a coverage of about 98% on all of these databases. It's important to note that with short-read sequencing technology, we cannot reach 100%. There are always regions in an exome that are too repetitive for short reads to cover. The second question is, are there other regions in coding genes that are of interest? And of course the answer is yes. Databases show now that we have more than 40,000 known variants that are pathogenic or likely pathogenic that lie outside of coding regions. ClinVar and HGMD, the two main databases, are being updated continuously. And if you combine them, you can see that ExomeXtra covers about 98% of these variants in the databases, while a vendor exome will not cover all of them. And the reason is simply that the vendor exome is not updated as often and cannot include all the newly discovered variants. Why are non-coding variants interesting? For an example, look here at a compound heterozygous variant situation in the ABCA4 gene. We have one variant that is in a coding exon on the right, and we have one variant that is in an intronic region on the left. And if we look at alignments generated from a vendor exome, an off-the-shelf exome, and from ExomeXtra, you can see that the second variant in the intronic region is not covered, because in the vendor exome we don't have coverage of this non-coding variant, and the case cannot be solved. With ExomeXtra, such cases can be solved. The next question is, are there maybe other types of genes besides coding genes that could be relevant? And again, the answer is yes. We know of non-coding genes, so-called RNA genes, that can also cause disease if they are affected by a variant. CeGaT ExomeXtra covers medically relevant non-coding genes. In an off-the-shelf exome, these will not be covered at all. Let's go to the next question. Are there any other genomes that are relevant? And of course, the answer again is yes, because the exome is just the human nuclear genome. And we have to also look at the mitochondrial genome, because important non-nuclear genes are in this genome, and again, variants affecting these genes can cause disease. This is why in CeGaT ExomeXtra we include the complete mitochondrial genome, and we sequence it at a higher depth to make sure that we can detect heteroplasmy. And the final question is, are there other genomic regions that are relevant? And it might not be a surprise, but again the answer is yes. Copy number variants, so-called CNVs, can defect the whole genome. And this is why in ExomeXtra we include a genome-wide backbone for CNV calling. CNV breakpoints in intronic and intergenic regions are usually detected using a method like array-CGH. By the inclusion of Xtra probes in these regions, we can now use the ExomeXtra for this detection. I will share an example with you on this next slide. You can see here we have a deletion that in a regular off-the-shelf exome we see as two separate stretches that we're not sure whether they are contiguous or not. And we are unclear about the breakpoint on both the left and the right end of this deletion. With the inclusion of the backbone in ExomeXtra, you can see in the lower part of the slide here that the breakpoint is very clearly resolved on the left and right, and we can see that this is one contiguous deletion. So, summing up, ExomeXtra provides comprehensive clinical exome coverage. We use up-to-date databases to define all coding exons of relevance. We include disease-causing non-coding mutations from current HGMD and ClinVar, as well as internal databases. We include clinically relevant non-coding genes, so-called RNA genes. We have pharmacogenetic variants included. We have the complete mitochondrial genome included. And we now have the whole genome backbone for CNV detection that gives you an array-CGH-like resolution for the CNV analysis. And very importantly, we regularly update all content to make sure that we stay current with respect to the scientific progress. And this is why we think CeGaT ExomeXtra is better than an exome and smarter than a genome. If you want to learn more about the ExomeXtra, please visit our website. And if you want to learn more about how we use this exome in clinical diagnostics, please have a look at our webinar from last summer where we show sample cases and discuss diagnostics in more detail. Thank you for your attention.